Hi, biology students. Thanks for watching this video of me reading today's reading and questions out loud. With that said, watch it at your own pace. Feel free to pause, think, answer questions, replay things, do what you got to do. Here we go. I'm going to begin reading in 3, 2, 1. Proteins and amino acids. What are amino acids? Amino acids are special organic molecules used by living organisms to make proteins. The main elements in amino acids are carbon, hydrogen, oxygen, and nitrogen. There are 20 different kinds of amino acids that combine to make proteins in our bodies. Our bodies can actually make some amino acids, but the rest we must get from our food. Questions round 1. What are proteins made of? Where do we get amino acids? What are proteins? Proteins are long chains of amino acids. There are thousands of different proteins in the human body. They provide all sorts of functions to help us survive. Why are they important? Proteins are essential for life. Around 20% of our body is made up of proteins. Every cell in our body uses proteins to perform functions. How are they made? Proteins are made inside cells. When a cell makes a protein, it is called protein synthesis. The instructions for how to make a protein are held in DNA molecules inside the cell nucleus. The two major stages in making a protein are called transcription and translation. Questions, round 2. What are bigger, proteins or amino acids? How do you know? What are the two major stages in making a protein called? Transcription. The first step in making a protein is called transcription. This is when a cell makes a copy or transcript of the DNA. The copy of the DNA is called RNA because it uses a different type of nucleic acid called ribonucleic acid. The ribonucleic acid, or RNA, is used in the next step, which is called translation. Questions, round 3. What is the difference between DNA and RNA? What is transcription? Translation. The next step in making a protein is called translation. This is when the RNA is converted or translated into a sequence of amino acids that makes up the protein. The translation process of making a new protein from RNA instructions takes place in a complex machine in the cell called the ribosome. The following steps take place in the ribosome. The RNA moves to the ribosome. This type of RNA is called the messenger RNA. It is abbreviated as mRNA, where the M is for messenger. The mRNA attaches itself to the ribosome. The ribosome figures out where to start on the mRNA by finding a special 3-letter begin sequence called a codon. The ribosome then moves down the strand of mRNA. Every 3 letters represents another amino acid molecule. The ribosome builds a string of amino acid bases on the codes in the mRNA. When the ribosome sees the stop code, it ends the translation of the protein, or it ends the translation and the protein is complete. Questions, round 4. What is mRNA? How many letters of RNA are in a codon? What in a cell are where in a cell are proteins built? Study this picture. Write 1 to 2 complete sentences describing the picture above. Study this picture. The picture shows protein synthesis. Find tRNA. What do you think tRNA is doing?